Today we're going to be talking about mitosis. And before we even get into what mitosis is, I'm going to show you a video. This is a video clip of a cell. Inside of this cell, you'll see these squiggly things. Those are chromosomes. Remember, chromosomes are made up of DNA, and although they look like worms and like they're alive, they're not living, okay? They're actually being pulled by strings and by like a skeleton within the cell. So as you can see, they're starting to line up in the middle. And then all of a sudden, they pull apart to either side. This is what mitosis is, okay? What happens in mitosis is we split apart the cells. And because the cells have DNA and chromosomes, those chromosomes and DNA have to be split in half as well. These are all just different video clips of cells dividing. So again, the squiggly guys, those are our chromosomes. You'll see them kind of form a line down the middle. They're lining up here. And eventually what we're going to see is we're going to see them being pulled to opposite ends of the cell. The cell in the middle here is going to pinch in, and now instead of one cell, we have two cells. And that is what mitosis is. So what we need you to do is go ahead and write down this title, Mitosis, in your notebook. We're going to go ahead and write down the definition of mitosis. So mitosis is the process by which body cells divide to produce new cells. You can see in the picture down below, we've got one cell, it's dividing and splitting into two cells. So this is the process. It's called mitosis. It happens in body cells, cells within your body. And this is how most of the cells in our body are formed, through mitosis. Now the key here is that mitosis only happens in what we kind of call our body cells. These are our stomach, our liver, our kidney, all the cells in our body except for our reproductive cells, so sex cells, sperm and egg. Basically, we divide cells into two main groups. We say body cells and sex cells, or body cells and reproductive cells. All of the cells in your body that are not reproductive are going to go through mitosis. The reproductive cells are going to go through a different process, and that will be our next unit. What I want you to do is see if you can think of another example of a body cell besides a stomach cell, a liver cell, or a kidney cell. You might have said a brain cell, or a heart cell, or a skin cell. Basically, any part of your body that you can think of, you can attach the word cell onto it, and it would work. Now, why do we need mitosis? Why do we need new cells? Well, our cells are wearing out all of the time. We need new cells in order to grow. As you're growing from a baby to a toddler to an adult, the number of cells in your body increases. That's actually what causes you to get taller. You're building new cells. So when you're a baby, you don't have as many cells as when you're an adult. Our cells also wear out. We get paper cuts. We shed dead skin, our hair grows, so we need to repair and replace our old cells. I like to give the example of even something as simple as just burning your tongue on some food. Well, those tongue cells don't stay burned forever. You eventually replace them with new cells. And this happens all the time. Your skin cells get replaced about every 7 days. Your hair cells are constantly growing. So mitosis helps us replace those cells. Now, mitosis makes two cells. We start with one, we call this kind of a parent cell, and it makes two cells. Here's what actually happens when we go through mitosis, is we start with 46 chromosomes. We copy the chromosomes, so now there's 92, and then we divide them in half. So our cells end up being an exact copy of our initial cell. So we start with 46 chromosomes, which is what our human cells have in each of them. We're going to end up with two cells, and they're each going to have 46 chromosomes. Think of mitosis as producing clones of cells. Okay, it wants to make an exact copy. If you burn your tongue and you need some new tongue cells, you want those to be the same type of cells as you had there before. You don't want them to be different. So mitosis makes kind of identical cells, exact copies of that initial cell. I'm going to have you write this down. It doesn't make a lot of sense right now, but go ahead and write down that the stages are what we call PMAT, P-M-A-T. Again, this is an abbreviation. We're going to talk about it in a couple slides, but just go ahead and write it down. So before we get into all of the parts of the cell dividing and how all this happens, we're going to talk about what the cell looks like when it's not dividing. So when the cell is not dividing, it's in a special phase. We call this interphase. Interphase means the cell's not dividing. Go ahead and write that down. You'll see on the right-hand side, this whole purplish gray area is interphase. So if this is a pie chart showing us how much time the cell spends dividing, you'll notice it doesn't spend very much time dividing. Interphase is when it's not dividing. That's a lot of time. And this green arrow is showing us mitosis, which is when it is dividing. So most of the time, cells are going to be not dividing. They're going to be doing their jobs. They're just going to be hanging out. But they do occasionally need to divide. Some cells divide more than others. So if we take a look at this, this is kind of like our pie chart from the previous phase. It says the orange part shows interphase. Don't worry about all of this, all the words. Blue part is mitosis when the cell is dividing. If you had to estimate the percent of time that the cell spends in interphase in this orange part, what would you guess? So the whole pie chart is 100. What would you guess the orange part is? A lot of people usually tell me, oh, probably something like 80 or 90. Yeah, anywhere around there would be totally fine. That's a lot of time it spends not dividing. So we need to talk about what happens when the cell is not dividing. So we already wrote down this top bullet point. We're going to write down the next two. When the cell is not dividing, the DNA is kind of all messy. We're going to say the DNA is in the form of what we call chromatin. Messy, uncoiled DNA. I like to think of this as just a big pile of yarn. Okay, that's kind of what DNA looks like. It's long strings, and it's not organized into chromosomes yet. Chromosomes are when it's nicely neat and organized. You'll notice we can also see the nuclear membrane. Okay, this nuclear membrane, it's just the membrane around the nucleus. I want you to make sure that you draw this picture next to interphase so you know what it looks like, because on the test, there's going to be pictures and you're going to have to identify what phase the cell is in. So if you have a picture here, it's going to help you. So we've got the scribbles of the chromatin, and we got the nucleus. Remember, this is when the cell's not dividing. Now we have a couple more pieces of information we're going to write down about interphase. This is the longest phase, okay? So we looked at this pie chart here and we said it spends a lot of time not dividing. So interphase is when it's not dividing. This is a really long phase. It spends a lot of time doing this. So if it's not dividing, what does it do? It's going to copy the DNA, it's going to grow, and it's going to copy its cell parts to prepare for cell division. Here we have a picture of cells underneath the microscope, and this green arrow is showing a cell in interphase. Notice when the cell's in interphase, we see the nucleus. Remember we said the nuclear membrane is visible? So this pink blob in the middle is the nucleus. I want you to see if you can find another example, the cell in interphase. You don't have to click anything, just see if you can find it. All of these are examples that I found, and there's even more over here. Notice they all kind of have that pink blob in the middle. Okay? So now that we talked about what the cell does when it's not dividing, we've got to talk about what it does when it is dividing. Remember, mitosis is when the cell is dividing. So stages of mitosis. We start with prophase. Remember I had you write down PMAT, P-M-A-T? Well, the P stands for prophase. That's the name of the first phase. So when the cell decides, oh, I need to divide, here's what happens first. Remember that messy chromatin, that DNA that just looked like a big pile of yarn? It's going to coil up all nice and neat. It coils up into chromosomes. A lot of times we draw these chromosomes as X's. So the chromatin condenses into chromosomes. I like to think of these X's as like nicely neat skeins of yarn that are all wrapped up. You know, when you go buy yarn right at the store and it looks like a nice, neat wrapped up thing. The reason the chromatin does this, it doesn't stay messy, is because we've got to be able to pull half to one side and half to the other. If you think about taking a big mess, tangled up mess of yarn and trying to split it in half, it's going to be really hard. So we got to package them up nice and neat so it's easy to split them in half. So again, we're going to draw this picture next to where we wrote prophase. You'll notice I've got a dotted line here for the nucleus. That's because in prophase, the nuclear membrane starts to disappear. It starts to dissolve. And this is so our chromosomes can move around. Remember, the DNA usually is stored within the nucleus. But we need those chromosomes to be able to move to either side of the cell so the cell can split. So this is our nuclear membrane. We've got our X's, which are our chromosomes. And we also get something called spindle fibers attaching to the chromosomes. Now, we have a lot of big words in this unit. Okay, scientists like big words. They like fancy names for stuff. So remember when we watched the video and we saw the chromosomes move? They didn't move on their own. Chromosomes can't move. They're not alive. They're just made of DNA. Okay, so what happens is these little strings are going to pull them. Those strings are called spindle fibers. And they're going to attach to the center of the chromosome. So a lot of times I tell students to draw this picture. Draw an X label it your chromosome. And in the middle, that's your centromere. That's the center of the chromosome where the chromatids, or the spindle fibers are going to attach. I know this is a lot of info. I know it's a lot of words, but let's take a look at it. Spindle fibers. What are they? You'll notice here we got our X's, our chromosomes. With little fibers, those little strings are going to be pulling them to each end of the cell. Okay? So if we take a look at prophase here, you can see an example of a cell in prophase. How do we know a cell's in prophase? Well, if we look back here, right, the chromosomes are in X's. Well, they're not going to look like that under the microscope, but we are going to be able to see those squiggles that look like chromosomes. The squiggles aren't going to be pulled to either side of the cell. They're not going to be lined up. It's just going to look kind of like a bunch of spaghetti all jumbled up in a circle. So think about where another cell that looks similar to this one is in this picture. I found this one. Sometimes people circle these. These are coming up later on because they're already being pulled apart or lined up at the center. All right, so P, prophase, that's the first phase. Remember we said P mat, so M is next. M stands for metaphase. This is when chromosomes line up at the center of the cell. Meta sounds like middle. Meta and middle both start with M. This is when the chromosomes line up in the middle of the cell. So for our picture then, we're going to draw. These are our little spindle fibers, right? Attaching. And we're going to draw the X's, the chromosomes in the middle. They're lining up. The reason they line up is so half can go to one side, half can go to the other. If we take a look at our picture, this is a different picture, but it's still cells under the microscope. Here we see chromosomes lined up in metaphase. Can you see if you can find another one that shows chromosomes lined up in the middle? Yeah, here are some more. So we see the squiggly chromosomes lined up in the middle. So we said prophase, metaphase. We said P mat, which means A comes next. A is for anaphase. What happens in anaphase? Well, the chromosomes start getting pulled to either ends of the cell. You'll see that they don't really look like X's anymore. That's because half of the X is going to one side and half of the X is going to the other. So they split up the DNA or the chromosomes equally. So when we're looking for cells, we're looking for ones that have the chromosomes starting to pull apart, like I have here. See if you can find one, chromosomes pulling apart. Here's an example of one. So PMAT, prophase, metaphase, anaphase, we're down to the last phase, which is telophase or telophase. People pronounce it differently depending on who you're talking to. I don't care which one you say. This is when the cell starts to pinch in. It's going to divide into two here. Okay, so it starts to divide. And our nucleus is going to reform. So again, you can go ahead and draw that picture. PMAT, prophase, metaphase, anaphase, telophase. Sometimes you'll hear people talk about cytokinesis. That's the last one, C, and that's where the cell completely splits in half. So if we look at telophase here, I'm just going to show you an example of one. Sometimes it's hard to identify. Here you can see the chromosomes at either end of the cell. So what I want you to do, just to review here, we're going to number these from 1 through 5, showing the correct order. So I'll tell you which one is first. This one's 1. Okay. So I want you to tell me, pick on the right-hand side, the choice that shows us the correct order. You should have gotten 2, 5, 1, 3, 4. So the first one here, okay, interphase, we've got our chromatin. Chromatin condenses into chromosomes, and we got prophase. Okay. Metaphase, they line up in the middle. Anaphase, they start to pull apart with those strings. And telophase, it splits into 2.